We often talk about medicine as if it's a sniper rifle. One shot, one very specific target.
Yeah.
You take a pill, it goes in, finds the problem, and boom, it fixes it.
That's the ideal for sure.
But the reality of pharmacology, it's often more like dropping a really important letter into a citywide mail system.
Mm-hmm.
You know the letter was delivered. You know the message was received because you see the reaction, but you have absolutely no idea which specific postal worker carried it, which truck it was on, or the route it took.
That is the fundamental black box of modern drug development.
Hmm?
We are very good at identifying the molecule, the drug itself, and the protein it's supposed to target. But biological tissue, it's not a uniform block of clay. It's a mosaic.
Right, and this is the concept of cellular heterogeneity.
Exactly. Even within a single tumor, or say a tiny slice of the brain's prefrontal cortex, you have this chaotic mix of different cell types. You might have 10 different subtypes of neurons sitting shoulder to shoulder.
And one responds to the drug.
One might respond beautifully, and its neighbor, which looks almost identical, might just completely ignore it.
And figuring out which neighbor is actually doing the work has historically just been a logistical nightmare.
It's a massive efficiency problem. Traditionally, to figure this out, you need what's called a paired data set. You have to take a tissue sample, treat it with the drug, and then you have to compare it to a control sample that you didn't treat.
The classic before and after photos.
You need both to spot the difference, which means you need to be able to treat the tissue in a lab, which is expensive, it's time-consuming, and frankly, sometimes it's ethically impossible if we're talking about, human brains.
Okay, which brings us to the breakthrough we're digging into today. This is a paper from Cell Reports Medicine by Li and colleagues, and they've developed a computational method called scRank.
That's right, scRank.
And the claim here is it's substantial. They're saying they can predict which cell types will respond to a drug using only the data from untreated tissue. No after photo needed.
It's basically predictive engineering for biology. Think of it like a structural engineer looking at the blueprints for a new bridge.
Yeah.
They don't need to wait for an earthquake to know where the stress fractures will appear. If the blueprint is accurate enough, they can calculate the collapse. scRank is trying to do that for our cellular networks.
So let's get into the mechanics. How does it actually work? The paper breaks it down into 3 phases, and phase 1 is network construction.
Oh, blueprint fair.
And the raw material for this blueprint is single-cell RNA sequencing, or scRNA-seq.
Right. Now, scRNA-seq gives us a parts list for every single cell, which genes are turned on and, how much. But a list of parts doesn't tell you how the machine actually runs.
It's just a list.
Exactly. So scRank needs to understand the relationships. It builds what's called a gene regulatory network, or a GRN.
So this moves us from gene A is present to gene A regulates gene B. But with 20,000 genes in a cell, that's a lot of potential connections. How does it filter out the noise?
Well, it's smart about it. It doesn't look at everything. It filters for the top 2,000 highly variable genes, the ones that are actually doing something dynamic. And it also looks at transcription factors, which are like the managers of the cell.
And it anchors all of this with known drug targets.
Yes, from databases like DGIDB. It uses what's already known to ground the map in reality.
I do want to drill down on the math here for a second, because the paper mentions principal component regression and tensor decomposition. These aren't just buzzwords. They're doing something really specific to clean up this map.
Oh, they're crucial. Think of it like a crowded cocktail party. Everyone's talking at once. If you just stick a microphone in the middle of the room, you just get white noise.
Right.
Principal component regression is a statistical method to isolate the independent conversations. It handles this big problem called multicollinearity, where lots of genes tend to move up and down together.
And the tensor decomposition part.
Think of that as the ultimate noise-canceling headphones. It takes this messy, high-dimensional data structure, and it sort of decomposes it to strip away all the background static, leaving you with a clean, weighted map. A map where the nodes are genes and the edges are the strength of their interaction.
So at the end of phase 1, we have a clean wiring diagram for every single cell type. Then we get to phase 2, which is where the action happens. In silico perturbation?
In silico, meaning in silicon, or, on the computer. This is the simulation part.
This is where we fake the earthquake.
Precisely. Let's say we're studying a drug that's an inhibitor. It blocks a specific gene. The algorithm goes into that network, finds the target gene, and sets its edge weights to 0.
It just mathematically cuts the wires?
It completely deletes the influence of that gene. And this creates what the authors call a target-perturbed GRN, or TPGRN.
I found their airport analogy really helpful here. If the gene network is a map of global air traffic and the drug target is a major hub like Chicago O'Hare,
then the simulation is a massive snowstorm that just shuts O'Hare down completely. No flights in, no flights out.
which leads us to phase 3. Perturbation evaluation. We have to measure the fallout from that storm.
Right. We have to see how much the system actually broke. So the algorithm compares the sunny day network, the untreated one, with the snowstorm network, the perturbed one, and it's looking for 2 specific types of damage.
Global and local.
Exactly. First, the global effect, and it measures this with something called manifold alignment.
Let's define manifold for everyone listening.
Sure. In this context, just imagine the gene network is a complex 3D shape, like a crumpled up ball of paper. Every crease and every fold represents a relationship between genes. Manifold alignment measures how much the overall geometry of that ball distorts when you cut those specific wires.
Does the whole structure just warp and change shape?
Exactly. And then there's the local effect.
Which is calculated with network diffusion.
Yeah, right. And that's asking, how far did the shock wave travel? When we shut down Chicago O'Hare, did it just delay flights in Milwaukee? Or did it cause cancellations in London, Tokyo, and Dubai?
And scRank basically argues that the cell type where the network collapses the most, where that shock wave is the biggest, that's the cell that's going to respond to the drug.
It ranks them. It says cell type A is structurally dependent on this gene. Cell type B barely even notices it's gone.
Okay, that is a lot of theory. Let's pause for a quick recap before we jump into the case studies to see if it actually works.
Heck of an idea.
All right, so recap number 1. The goal here is to find which specific cell type responds to a drug, but without actually treating the tissue.
How predictive.
The input is just untreated single-cell data, plus the name of the drug's target gene. The process has 3 steps. 1, construct the network, using that fancy math to build a clean wiring diagram, the GRN.
Step 1, build the blueprint.
2, perturb. Simulate the drug by mathematically cutting the wires to that target gene.
The in silico earthquake.
And 3, evaluate. Measure the chaos, both globally with manifold alignment and locally with network diffusion. The output is just a ranked list of cells from most responsive to least responsive.
That's a perfect summary. The core logic is that structure predicts function. If the network structure relies on that target gene, the cell itself must rely on it too.
But biology just loves to ruin elegant theories. So let's look at the proof. The paper details a fascinating case study on major depressive disorder, or MDD.
This was a critical test. I mean, we treat millions of people with SSRIs like fluoxetine, Prozac. We know the target molecule is the serotonin transporter, which is encoded by the gene SLC6A4.
So we know the drug, we know the gene. But the brain is the most heterogeneous tissue in the entire body.
And that's the knowledge gap. We didn't know exactly which subtype of neuron was the primary responder. Is it all of them? Is it just the sensory neurons? Who's doing the work?
So they fed scRank data from untreated brains of patients with MDD. They told the computer the target is SLC6A4. What did it find?
It pointed with incredible specificity to one particular cluster, X9, which stands for Excitatory Neuron Cluster 9.
Why X9? What was so special about its wiring diagram compared to, say, cluster 5 or another neuron?
Well, it comes right back to that topology we were just talking about. In most of the other cells, like inhibitory cluster 5, the SLC6A4 gene was present, but it was a satellite. It was on the periphery of the network.
Okay.
So cutting it was like closing a tiny regional airstrip. Traffic just rerouted. No big deal.
But in X9?
In X9, SLC6A4 wasn't a satellite. It was a hub. It was the central node of a highly active functional module 2 specifically. It was connected to everything. So when you cut it, the entire local network collapsed.
A computer prediction is one thing, but did they verify this in the physical world, in an actual brain?
They did, and the validation was really multi-layered and elegant. First, they looked at startle data. They found that these X9 neurons are located in the deep layers of the cortex layers 5 and 6. Then they analyzed gene expression in actual MDD patients, and they found that those specific deep layer neurons showed abnormally high activity in serotonin-related genes compared to healthy people.
So scRank didn't just find a cell that could respond. It seems like it identified the very cell type that was driving the disease state.
It pinpointed the precise vulnerability in the entire system.
Let's move to the second case study, because this one involves a mechanism discovery that I found really, really impressive. We're moving from the brain to the heart myocardial infarction, a heart attack.
Right. And specifically, the drug is Tanshinone IIA. It's a compound from traditional Chinese medicine that's known to reduce inflammation after a heart attack.
Okay, so they ran the algorithm on cardiac macrophages. These are the immune cells that clean up the damage in the heart.
And scRank predicted that macrophage subtype 5, or MB5, was the top target. But here is where the story gets really interesting.
The twist.
The researchers already knew that Tanshinone IIA targets a gene called CTSB. But when scRank simulated the network shutdown in these MB5 macrophages, it noticed a massive ripple effect spreading to a downstream gene, a gene called cathepsin B, or CTSB.
So the algorithm predicted a domino effect. It said, look, if you hit 6 gig, you're also going to knock over CTSB.
Exactly. And this was not a well-established mechanism for this drug at all. It was a purely computational inference.
So they had to go to the wet lab and actually prove it.
They did, and they used this fascinating technique called the cellular thermal shift assay, or CETSA.
Could you explain that for us?
Yeah, it's really cool. It's based on a simple principle. When a drug binds to a protein, it makes that protein more stable. It makes it more resistant to heat. So they literally heat the cells up. If the protein survives at higher temperatures, it means the drug must be bound to it.
Did it work?
It did. They confirmed that Tanshinone IIA effectively binds to and reduces the expression of the CTSB protein, just like the algorithm predicted.
So scRank isn't just a GPS for finding the right cells. It can be a mechanism discovery tool.
It elucidated a whole molecular pathway for a traditional drug just by analyzing the shape of its network connections.
Okay, let's hit our second recap here because that really covers the applications. So first, the brain case. scRank used untreated data to pinpoint X9 deep layer neurons as the target for antidepressants because the target gene was a major hub in their specific network.
The linchpin.
Then the heart case. It identified MB5 macrophages for a heart attack drug. But the bonus here was that it predicted a downstream effect on a totally different protein, CTSB, which was then experimentally proven.
A new mechanism discovered by the algorithm.
So the big takeaway is that the algorithm seems to work across completely different tissues, brain versus heart, and different drug types, synthetic versus natural.
It seems to be very robust.
So why does all this matter? I mean, beyond writing cool papers, what is the so what for a patient or for a pharmaceutical company?
I think the number one implication is precision medicine. We are really moving away from the era of this drug targets protein X. We are entering the era of this drug targets cell type Y in patient Z.
Because my cell type Y might be wired completely differently than yours.
Exactly. If your network topology is different, if that gene isn't a hub for you, the drug might not work, even if you have the target gene. But there's a massive economic angle here, too, drug repurposing.
Right. We have these giant public databases like the Cancer Genome Atlas, TCGA.
Which are just mountains of untreated biopsy data. We rarely have the treated data because running clinical trials where you take biopsies after giving an experimental drug is dangerous, it's expensive, and it's rare.
So scRank basically unlocks the value of all that existing data.
You can take a drug that failed for one disease, run its target through scRank against a database for a totally different disease, and see if it hits a crucial cell type. It's like finding a new key for a lock you've had sitting in a drawer for 20 years.
We should address the limitations, though. I mean, scRank isn't perfect.
Okay. No model is. And the authors are quite transparent that scRank currently models inhibitors best.
Because the simulation involves cutting wires.
Right. Deleting an edge from the network is a great way to mimic blocking a gene. But what if your drug is an agonist, a booster? Modeling extra connection strength is mathematically much, much trickier than modeling a disconnection. They did some testing on it, but its strength is definitely with inhibitors right now.
And what about performance? Did they benchmark this against the old school methods?
They did. They compared it against tools like Augur and standard differential expression analysis.
But those methods usually get to cheat, don't they? They get to see the post-treatment data.
That's the thing. scRank was basically fighting with one hand tied behind its back, using only the pre-treatment data, and yet it either beat them or matched them in almost every scenario.
Even when the cells were really similar to each other.
That's the real kicker. Even in datasets with low cellular heterogeneity, where the cell subtypes look almost identical on the surface, scRank could tell them apart based on their internal wiring.
So it proves the information wasn't just in the expression levels, how loud the genes are shouting. The information was hidden in the topology, who they were shouting at.
Topology is the key word of this whole deep dive. We are entering an era where biological networks are telling us more than just lists of parts. The shape of the connections is what defines the function.
Before we wrap up, I want to explore a thought that is Well, it's both exciting and a little bit terrifying.
Gone.
If we can predict which cells will respond to a drug based on their network, could we predict side effects?
That is the provocative question, isn't it?
Okay, so say I have a new cancer drug. scRank says, great, it targets the tumor cells perfectly. But what if I run scRank on a database of healthy liver cells or kidney cells, and I see that the target gene is a major hub there too?
Then scRank would, in theory, predict that the healthy network would also collapse. You've just predicted a toxic side effect.
Before a clinical trial even starts? Before a single animal model is used?
Potentially, yes. We could screen drugs for toxicity entirely in silico, saving billions of dollars and, more importantly, avoiding dangerous trials for patients. If a healthy cell has a vulnerable network topology, this tool should be able to flag it.
That's a massive shift in how we think about drug safety, moving that fail-fast moment from the clinic all the way back to the computer.
It creates a safety net based on pure math.
So here's our question for you listening today. We often trust our doctors, and we trust the FDA. But imagine a future where you have your own biodigital twin, a complete map of your personal cellular networks. If scRank ran a simulation on your biology and said, this drug will work for you, but the general clinical trial data said it only works for 30% of people, who would you trust? Would you trust the algorithm?
It's that age-old question of trusting the map versus trusting the territory, but these maps are getting surprisingly good.
Thanks for diving deep with us.
See you next time.